Today in this video we will discuss about this competitive inhibition, which is part of the reversible inhibition. So as the name implies, reversible, it means that inhibitor will bind to enzyme and at the same time it can easily be removed. So if enzyme binds to inhibitor, in that case enzyme can also get rid of this inhibitor, and we will discuss about one of these reversible inhibition, which is competitive inhibition. So as the name implies competitive, it indicates that there is a competition. So competition between what? Competition between inhibitor and substrate. So before going into discuss about all those things, let us discuss about the basic process of enzyme and substrate complex formation as well as product formation. So what happens is that enzyme active site allows the binding of the substrate molecule. The shape of the substrate is very much complementary to the enzyme active site, which helps them to bind to the enzyme active site, that allows the formation of enzyme substrate complex followed by the formation of product, which you can see here. So this one is the binding of substrate to the enzyme, and this one is a formation of enzyme substrate complex, and here the product is formed and it gets released. So enzymes are basically biocatalyst, and inside our body there are lots of biological reactions take place, and this biocatalyst enzyme speeds up the rate of reaction. Now the question comes, what is the role of inhibition here? So if you look at this particular figure, you are seeing that the reaction ends with the formation of product. So the formation of product is highly regulated inside the cell. That means there is a sufficient amount, there is a concentration above which this product is not needed, or the body wants to get rid of this product because there is a certain amount that the cell demands for, and this regulation is achieved with the help of several ways. The one is the expression of enzyme. So if the expression of enzyme gets regulated, in that case the formation of product will also get regulated. What that means is that if the expression of enzyme gets lower up, it won't be available to form this product. Okay. So if the expression is completely shut off, no enzyme will be produced and no product formation is there. And another one, if the expression is there but the activity is not there, in that case also product will not form. So again it means that if we will inhibit the activity of this enzyme, then in that case also the product formation will not take place. So these are the two different ways. So there are certain molecules, there are certain ions that binds to this active site of enzyme and thereby inhibits the chemical reaction to inhibits the formation of product. So if you look at this figure of competitive inhibition, as I have said you earlier, that in this type of inhibition there is a competition. So when the term competition comes, it indicates that there might be a certain kind of similarity. So the similarity in this case is between the substrate and inhibitor. So the inhibitor is very much similar in structure to substrate, and this inhibitor also has a very higher affinity for this enzyme. So as a result of which, in this competition, inhibitor wins and substrate get out. So at the end inhibitor will bind and the enzymatic activity will get lessen up. Okay. So what that means is, if the amount of inhibitor get lessen up and if we increase the amount of substrate, then in that case what will happen, inhibitor is less likely to bind to enzyme active site. So let us suppose there are 5 substrate molecule. Okay, so there are 5 substrate molecules and there are 5 inhibitor. So because the affinity of inhibitor is very high, in that case inhibitor will bind to the substrate. Now if you increase the amount of substrate molecule to say let's 20, in that case what will happen, there is chances that 5 of the substrate will bind here there is chances that because the availability of substrate is very high than inhibitor, so there is chances that almost all the time the reaction is occurring, the substrate is going and binding to the active site because the concentration of substrate is very high. So this is what happened in case of competitive inhibition. Just because both this substrate as well as this inhibitor binds to the same site, so if you increase the concentration of this substrate, there is a lesser chance for this inhibitor to come and bind to active site, because every time enzyme will have the opportunity to bind to the substrate. Okay, and because the concentration of inhibitor will be very less, it will also have a less chance to encounter the enzyme active site. So if you look at this figure, so this figure shows that enzyme binds with substrate to form enzyme substrate complex, and finally product is formed and it gets released from the enzyme. So when there is no inhibitor is there, then this is what happens. Okay, so this is the general scenario that occurs in the absence of inhibitor. But what happens when the inhibitor is present, when competitive inhibitor in general is present, when this inhibitor is present? Just because I have said you that there is a competition, because both the substrate as well as inhibitor, both of them are highly similar in structure. So in that case what happens, the inhibitor, because it has higher affinity, because it will bind the enzyme active site, so it outcompetes the substrate and forms the enzyme inhibitor complex. So the only way to get rid of this inhibition is if you increase the substrate concentration more and more, there is more chances that substrate will bind to the enzyme and enzyme substrate complex will form. So if you look at the reaction kinetics of this competitive inhibitor with respect to the condition when there is no inhibitor is present, then in this case you can see that Km is increased while Vmax is constant. So how does this Km is increasing? So you know that Km is that substrate concentration at which V0 is half of Vmax. So if you look here, if you look at this figure, when there is no inhibitor, in this case this is Vmax, this axis is, this line is denoting Vmax, this is the rate of reaction of V. So and this axis denotes the substrate concentration. So you can see that when there, you first look at this curve. So when you see here that Km is that substrate concentration at which the V0 is half of Vmax. So when you look at the half of Vmax, when you see at this line is that point where V0 is half of Vmax, and Km is that substrate concentration. That this means that this is the axis where substrate concentration is coming. So this axis denoting substrate concentration, and since Km is that substrate concentration, that means Km tends to localize on this axis as it represents the substrate concentration. So Km is that substrate concentration at which V0 is half of Vmax. So this is the point where V0 is half of Vmax. Okay, so at this point V0 is half of Vmax and Km is that substrate concentration at which V0 is half of Vmax. Now what happens when competitive inhibitor is present? When competitive inhibitor is present, in that case we are increasing the concentration of substrate. Since we have to increase the substrate concentration to reach the same Vmax, in that case what you will see that again, if you look at this point also, V0 Km is that substrate concentration at which V0 is half of Vmax. So again, if you look at this point in, and you see that when you merge these two axes at this point, when you see that when you bring this half of Vmax line to this curve of competitive inhibition, and when you go down to match up with the substrate concentration, which will indicate the Km value, in that case you will see that the Km has been increased from this to this. So you will see that Km is also getting increased. However, Vmax and V0 for both the graph is same. Okay, so if you see that Vmax is same here, and half of Vmax is also same for the this part and this part. So only one thing is getting changed, that is the Km. So in case of competitive inhibition, there is no net change in the Vmax value, but there is an increase in this Km value because the amount of substrate concentration is we need to increase to reach that particular Vmax value. So for us to reach that particular Vmax value, we need to increase the substrate concentration, and you can see that from this point only that in spite of this point same for both the control as well as the inhibitor part, but the Km is different because to reach the same Vmax we need a higher substrate concentration, and in this case for competitive inhibitor this Km is higher because Km represent that part at which V0 is half of Vmax, and in presence of this competitive inhibition the Km is higher, so the Km gets increased in case of competitive inhibition. So in the next few video we will discuss about the non-competitive, uncompetitive as well as irreversible inhibition. And thanks for watching this video.